Hello students, welcome to this session. So from now onwards we will be having discussion on the topics. So first is hemoglobin. The methods of hemoglobin estimation. So the first one is Siley method. The principle is acid hematine method. The second is calorimetric method. The principle is based on cyanamide hemoglobin method. Then it can be gasometric hemoglobin by calculation oxygen carrying capacity, or chemical method like acid hematine or alkaline hematine, and specific gravity method by use of copper sulfate. So it is mainly used in blood bank for donor screening. So for Siley method, the Siley apparatus is used, that is hemoglobinometer. It consists of hemoglobin pipette. So for that 0.5 marking, which equal to 20 microliter of the blood. Hemoglobin tube for dilution of 0.1 NHCl and hemoglobin, and comparator, that is brown glass present in Siley apparatus for the comparison of that hemoglobin acid dilution, and the use of hemoglobinometer is for the hemoglobin estimation. The principle of Siley method. So when we add blood to 0.1 HCl hydrochloric acid, hemoglobin is converted to brown color acid hematine, and then that acid hematine is diluted with the distilled water till we get the matching of that dilution with the standard brown glass, that is Siley's hemoglobinometer. So now we will see the procedure of hemoglobin estimation by Siley method. So for that the sample is capillary blood or the EDTA blood. So with help of Pasteur pipette we have to take 0.1 NHCl in hemoglobin tube up to 20% mark. Then blood we have to take in HB pipette or Siley pipette up to 0.5 marking or 20 microliter blood, so that there should not be air bubble and excess blood should be wiped out with the dry cotton or cloth. Then that blood is transferred to the hemoglobin tube which already contains 0.1 HCl. So now we have to mix and then wait for 10 minutes, and then after that we have to dilute that solution by adding distilled water drop by drop and mix, and then see the matching of that dilution with the matching of brown color plate or comparator, and when it gets matched then the value of that tube gives you the hemoglobin concentration. So the principle is when blood is mixed with Drapkin's reagent which contains potassium ferricyanide and potassium cyanide. So hemoglobin reacts with potassium ferricyanide and gets converted to cyanamide hemoglobin HCN by cyanide, and then intensity of that color is proportional to hemoglobin concentration, and it is compared with known hemoglobin standard, that is cyanamide hemoglobin standard at 540 nanometer, that is green filter used in the calorimeter. Now we will see the procedure. So for that sample it could be capillary blood or anticoagulated EDTA blood, so that we can take as a estimation. Then we have to take two test tubes, one test tube labeled and then one label as blank tube. So now we have to add 5 ml of Drapkin reagent to each test tube, 5 ml in test tube and 5 ml in blank tube, and then 0.02 ml or 20 microliter blood we can take with help of Siley pipette or auto pipette, that we have to add into the tube labeled as test. Then mix and wait for 5 minutes, and then read the absorbance of that test tube by setting the blank with the blank tube, and then read the absorbance of standard which is already provided in the kit. It equals to 15 gram%, and then we have to calculate the concentration of test or unknown by using a formula. So the formula for unknown concentration of the hemoglobin is OD of test, that is optical density of test divided by optical density of standard, and then we have to multiply it by concentration of standard, that is 15, and then we can write the result. The hemoglobin concentration of the given blood sample by Drapkin method or by calorimetric method is dash dash gram per dl. principle is blood is diluted 1 to 200, that is 1 part blood and 200 part RBC diluting fluid. So when it is diluted 1 to 200, and then cells are counted under high power by using Newborn's chamber. So in that RBC diluting fluid is used, and it is isotonic with blood, so hemolysis of the RBC do not take place, and sample is capillary blood or EDTA blood. Procedure: so you have to take 0.5 mark in the RBC pipette blood, and then dilute it with RBC diluting fluid up to 101 mark, then mix and then discard few drops, and then load the chamber, wait for few minutes, and then see the RBCs. So in that the central square is RBC counting chair, and in that you have to count total number of RBCs, and the calculation is total RBC count is equal to n into 10,000. So in that method is hemocytometry or by using Newborn's counting chamber. Principle here, the dilution is 1 to 200. So in that platelet diluting fluid is used, which contains procin hydrochloride, and then it lyses RBCs, and then you can see only platelets. Sample is EDTA or anticoagulated blood. Procedure in short: up to 0.5 mark in RBC pipette you have to take blood, and then up to 101 mark you have to take platelet diluting fluid, then mix, discard few drops, then load the chamber and count the platelets. So counting of that platelets is done in the central RBC counting or platelet counting square. In that corner 4 squares and central 1 square is counted, or total you can count all the squares, and then formula is total platelet count is equal to n into 2000. In that method is by hemocytometry or by use of Newborn's chamber. So principle is, in that WBC diluting fluid is there, which contains glacial acetic acid, which lyses the red blood cells, and gentian violet, which stains the nuclei of the white blood cells. And then when you do dilution 1 to 20 in WBC pipette, then and then see under high power, you will see only WBCs, and then you can count that. And sample used is anticoagulated, that is EDTA blood or capillary blood. So procedure in short: take 0.5 mark, take blood up to 0.5 mark in WBC pipette, then dilute it with WBC diluting fluid up to 11 mark, mix, wait for a few minutes, then load the chamber and count the WBCs in 4 corner of the WBC area, that is 4 corner of the Newborn's chamber, and then do the calculation by formula. Total WBC count is equal to n into 50. So principle, in that we can use Leishman stain, Giemsa stain, field stains. So they are all polychromatic stains. So in that methanol acts as a fixative, which fixes the blood smear and do not change the cells. Then basic component of the white blood cell, that is cytoplasm, is stained by acidic dye, and then it is seen as eosinophilic. And acidic component, that is nucleus of the cell, is stained with basic dye, and then it appears as blue or violet in color, and they are called as basophilic. So in the procedure short, you have to take dry clean glass slide, then take 1 drop of blood at the end of the slide, and then with the help of another spreader slide you have to spread that drop forward in shape of tongue, and then you can stain it, fix and stain it. So staining by Leishman method. So dry the smear, then put it on the glass rod, then overflow the stain over the slide, that is Leishman stain. Wait for 5 minutes, then dilute it with buffer, wait for 5 minutes, and then wash with tap water, and then dry the smear, put oil, and then see under oil immersion lens, and then see the morphology of the WBCs. Absolute eosinophil count, it is blood is diluted with specific diluting fluid, that is absolute diluting fluid, which removes red blood cells and stains eosinophils red in color. These cells are then counted under low high power by using Newborn's counting chamber. Procedure: take 0.36 ml of diluting fluid in test tube, add 0.04 ml of blood by using HB pipette twice, mix and keep for 10 minutes. Mix the diluent, load the chamber, and then allow to wait for 2 to 3 minutes, and then count the cells under low power. So calculation: total number of eosinophils per cubic millimeter can be number of cells counted in the 9 squares into 10 divided by 0.9. Result: it is the absolute eosinophil count given in the blood sample. Reticulocyte count: so it is supravital staining method. So in that, blood is mixed with the stain, that is brilliant crescent blue of the stain, and then it enters the blood cells in living condition. So RNA in the cells is precipitated by staining as dark blue network or reticulum, and then blood smear is made afterwards. Specimen: EDTA or capillary blood. So procedure: in test tube, take 2 drops of blood and 2 drops of stain, then mix thoroughly, then put that test tube into the incubator for half an hour, then prepare the thin smear of the preparation of the blood and stain, then air dry, and then see under microscope. So these reticulocytes are seen as they are green cells, then identified reticulocytes by deep violet filaments and granules in the network inside the green RBCs, that is reticulocytes. So calculation: reticulocyte count percentage 100, number of red blood cells counted, number of reticulocytes counted, and then percentage of that. So reticulocyte given in percentage. So usually reticulocytes are counted in the tail portion of the smear. So procedure is by 2 methods: Westergreen method or Wintrobe method. So long tube is called as which has marking 0 to 200, it is called as Westergreen tube, and Wintrobe tube is of 0 to 10 marking. So fill the Westergreen or Wintrobe tube and then wait, keep it in stand and then wait for 1 hour and then take the reading. So that is the procedure. So we will see procedure of PCV by Wintrobe method. So fill the Wintrobe tube with blood with help of pasteur pipette, then keep it in centrifuge. Take another Wintrobe tube filled with water and put it in the centrifuge opposite to the balance, to the previous tube for the balance, and then centrifuge it for 30 minutes and then take the reading of red cell column from the bottom of the tube. So peripheral blood smear: first is the sample collection. So it is a simple and gold standard confirmatory test for the malaria and filaria or other blood parasites. So specimen: the collected peripheral blood from the earlobe or by finger prick or great toe in the infants. The time of taking blood should be collected few hours after the height of the paroxysm of this fever and before taking antimalarial or antifilarial drugs. Frequency: smears should be examined at least twice daily until parasites are detected. The types of the blood smears: there are 2 types, thin blood smear and thick blood smear. So you can see in this picture. Preparation and staining of blood smears. So thick blood film, so it can be prepared by taking 2 to 3 drops in the center of the clean slide and then just make a round with help of another spreader slide or some another material. Then in thin smear, you have to take 1 to 2 drops of blood at the end of the slide, and then you can forward it by help of another spreader slide, and then you will get tongue shaped smear. So this is thin smear. After preparation of the smears, that is thick smear and thin smear, we can stain the slides either by field stain or Leishman stain. So first we will see field stain. So here we have to make the smear, dry it in the air, then fix the smear with help of methanol or the spray, and then dry it in air. Then you have to put that slide into the field stain jar B, and then for the 5 to 6 seconds wash it with the running water. Then put that slide into the container containing field stain A, which is blue in color. Field stain B is pink in color. So field A you have to put for 10 to 30 seconds, then wash, dry, put oil and see under oil immersion lens. So usually this field stain is done for the thin smear. So the second method is Leishman stain or Wright stain. So flood the smear with the Leishman stain for 2 to 3 minutes. Then dilute it with the equal volume of buffer water. Then wait for 5 minutes and then wash and see the smear after drying by putting oil into the oil immersion lens. So this is a stain smear. So the observation in thick and thin smear. So usually in thick blood film, you can see only the imprints of the parasites. That is the ring form blue in color and the nucleus is pink in color, and very few RBCs you can see. And thin blood smear, you can see plenty of RBCs containing the ring form of the parasites. That is thin nucleus is pink in color and the ring is blue in color. Now we will see difference between thick and thin smear. So usually thick smear is used for the screening purpose and thin smear is used for the identification of the species by different morphological forms. So thick smear, the advantages, it is more sensitive. When only 5 to 10 parasites per microliter of blood is there, you can see the parasites. And the quantification of the parasite, that is parasitic index, you can make out from the thick smear. And demonstrating malarial pigments like Schaffner's dots, Zeeman pigments in the different species of the malaria. The disadvantage of the thick smear, the species identification is not possible. The thin smear advantages, the species identification of the malaria is possible because of the different morphological forms, that is ring form, tropozoite, seizoid, gametocyte form of the species. The disadvantages, it has the low sensitivity, so you need more than 200 parasites per microliter of the blood to see the parasites. So these are different morphological forms of the parasites of the Plasmodium vivax. So first A shows ring form of the Plasmodium vivax. The ring is thick blue in color and the nucleus is pink in color. Then you can see the RBCs containing Schaffner's dots, that is pink color dots, and the amoeboid tropozoite of the P vivax, that is blue in color and the nucleus pink in color. Then you can see seizoid form in the C, that is many merozoites are there in the regular round structure, that is seizoid form. Then you can see male gametocyte and female gametocyte form of the Plasmodium vivax. Now you can see in A, that is Plasmodium falciparum morphological form. So in A you can see ring forms. So there are few RBCs containing multiple rings, few RBCs containing the headphone appearance of the ring form of the Plasmodium falciparum, or you can see a cold form. Then in B you can see banana shaped B and C you can see banana shaped gametocytes of the Plasmodium falciparum. So only two forms are seen in Plasmodium falciparum. Seizoid and tropozoite are usually not seen in peripheral blood. Then in D you can see ring form of the Plasmodium ovale. So RBC is oval in shape, so you can, because of that the name is Plasmodium ovale. And in E you can see band form tropozoite of the Plasmodium malariae. So uses of the peripheral blood smear for the blood parasites. So first is Plasmodium species, then Trypanosoma, then Leishmania, you can see LD bodies, then Wucheria bancrofti. So microfilarial forms you can see in the slide. So these are different parasites you can detect into the PBS. Uses of peripheral blood smear. So reporting can be done. First is RBC morphology. So we can see size, shape and inclusions present in the RBCs. So here few examples. So in size variation can be bigger RBCs, macrocytes, smaller RBC, microcytes, oval macrocytes, or according to the color of the RBCs, it is hypochromasia if it is pale in color. If it is purple in color, then it is polychromasia, or hyperchromasia if they are spirocytes. Then different shapes, it can be target cell, teardrop cell, oval cells, elliptocytes, tomatocytes, pencil shape, sickle shape, burr cell. So these are different shapes. And inclusions, some basophilic stippling can be present, purple granules inside the RBCs, ring form that is called Cabot ring, hinge bodies. So the Howell-Jolly body is seen in megaloblastic anemia. So these are different RBC morphology we can see. Then WBC we can report as total count, whether it is normal, increase or decrease, differential count, whether it is lymphocytosis, neutrophilia or eosinocytophilia. Then abnormal cells, any atypical cells, blast cells or other abnormal cells. Platelet count can be normal or abnormal, and then morphology, so normal in size or bigger in size. Then parasites like Plasmodium, Filaria, Leishmania, different morphological forms, ring form, tropozoite form, gametocyte form, seizoid form, or in Filaria, microfilaria, we can see. So this is reporting about PBS. These are my references from where I have taken my notes, and I hope you find this session very useful for your academics and for your future practice. Thank you so much.